Hello, this is a review for your midterm study guide for Biology 1001. You should have gone ahead and received a study guide on the Remind app as well as posted on Blackboard. I included some useful websites to assist you as well. The websites will provide you with video clips and other information to assist you in any topics that you may be unfamiliar with. I am going to discuss a few of these topics within this 15-minute review session. And if you have any questions, please feel free to email me or message me on Remind. The first thing I want to talk about are the differences between prokaryotic and eukaryotic cells. We know that there are various types of cells. For example, we have blood cells, skin cells, bacteria cells, all of which have different functions in the world around us. But one thing we want to talk about is our main differentiated structures of prokaryotic cells as well as eukaryotic cells. So we know that a basic cell structure is present, but not all living is the nucleus, which is the structure in the cytoplasm surrounded by a membrane, a nuclear membrane, and protects most of the cell's DNA. Now within a prokaryotic cell, if you look, you have circular DNA. It's free-floating DNA. It's not encapsulated inside of a nucleus. There is not a membrane-bound structure, but you do have your plasmid, you have your pili, which are going to help with movement, cytoplasma, the plasma membrane, that cell wall that's going to provide support as well as a capsule. The flagellum is going to be used for movement as well. So these are the things to remember, that a prokaryotic is going to be one of your basic cells, and that basic cells such as bacteria is a great example of a prokaryotic cell. Now with eukaryotic cells, of course, they contain a nucleus and membrane-bound organelles. Most times they are quite larger than prokaryotic cells and are found in multicellular organisms. So that's why they're called eukaryotic cells. And so they contain different organelles that have different functions within themselves. So we know, for instance, the mitochondria, a lot of times we call it the powerhouse of the cell. It provides energy. It's the site of cellular respiration. It's one of the eukaryotic organelles. And so they carry out many different functions. And even looking at this picture, you can see the complexity of a eukaryotic cell versus a prokaryotic cell. You see there are much more components that are found within a eukaryotic cell as opposed to a prokaryotic cell. One thing to remember is that there is no cell wall. So if you think back, recall, that was one of the earlier labs that we did, and being able to differentiate between the two. Here's a chart that I would just like to point out. Nucleus, no. Eukaryotic, yes. Yes, they both contain DNA. Cell wall always reasonably says dependent in a eukaryotic, because a plant cell has a cell wall, whereas the animal cell does not. And there is a cell membrane and membrane-bound organelles in eukaryotic and not in the pro. Ribosomes are in both. So these are the summary points that I just want to point out. Prokaryotic cells are cells without a nucleus. Eukaryotic cells are cells that contain a nucleus. And eukaryotic cells have organelles besides the nucleus. The only organelles in the prokaryotic cell are ribosomes. Take a moment. You may want to pause the video and see if you're able to answer the 5 questions. The next section I would like to talk about is classification with taxonomy. We did a lab with this one, and we know that we have a variety of different organisms on Earth that range from a small, simple cell to a very complex mammal. So we use taxonomy to classify these things into groups. Tax is plural, and singular is taxon. So we know the kingdom is a major grouping organism of the same type. So we know we have Homo sapiens. That's where we're describing that genus and that species, and the 2 naming system for the nomenclature of this. So here we are looking at hierarchy of classification. Just a simple example, this is a hierarchy of classification of cars. And so we have size, color, number of doors, and the seats. And so we know that when we classify, that is going to give us an understanding of where various organisms belong. And so this is a summary that taxonomy is a scientific classification of organisms, and they classify them to make sense of tremendous diversity on Earth. So you may want to be able to explain taxonomy, why biologists need to classify. We know that we classify in order to be able to define which organisms belong within a specific group, so that when we're studying for various types of diseases or just to get a better understanding of organisms, we know which ones fall within certain categories and why they have similar characteristics. And so, what is the most diverse group of animals? I want you to think about that. And so, if you want to, you can create a hierarchy taxonomy to classify writing implements such as pens and pencils, if you so choose, just to get you some practice. So when we are naming, we have to name in the system of naming nomenclature, and we know that it is binomial. And so it's a binomial system.
That's its genus. It's written with a capital letter at the start, and it's written in italics. See the second name? That's its specific epithet, which is a fancy way of saying that it refers to one species in the genus. It has a lowercase letter, and it's also written in italics. So why do we care about these scientific names? Well, you could come up with a lot of common names for an organism that vary based on location. Take this mountain lion, for example. It's also known as a puma, cougar, or Texas panther. Same animal, different names. But its scientific name here is specific and recognized regardless of your location. And that gives power to an awesome way to organize and name species.
We know that when we break it down, we go by domain, kingdom, phylum, and we just continue on. We have subphylum and superclass, but we didn't break those down. But in a little more detailed biology classes, you may see that class, subclass, order, family, genus, and species. So just focusing on the classification name, remember we said King Philip came over for good spaghetti. And that will help you in remembering the order in which the classification goes. And as you can see, you know your domain as you carry it here. There's only 3 main domains. Then we break it down to our various kingdom, and we go through that process. This is one thing that was simple, red maple. Refresher on body symmetry. We know we had radial symmetry, bilateral symmetry, sagittal divides the body into right and left halves. We didn't really focus on that one, but we did focus on looking at bilateral symmetry, meaning that we could cut it down the middle, and it's basically a mirror image of each other, resulting in 2 mirror images of right and left halves, such as those of a butterfly, crab, or a human. When we look at the echinodermata, such as sea stars and sand dollars and sea urchins, they display a radial symmetry as adults, but their larval stages exhibit bilateral symmetry. And so the term is secondary radial symmetry, and they believe to evolve. So we said we can cut those and in any way. That's why it's radial. Want to focus on the different types of bacteria shape. We have coccus, bacillus, and spirillum. So these are pictures. Make sure you're able to identify the various types of bacteria based off their shape. So their names really could pretty much go with their shape. So spirillum, kind of think of like spiral cocci, you know, the circular. Here's your bacillus. And so just make sure that you are able to remember this. Remember reproduction, bacteria produced by fission, process that gives you 2 new cells. And then we know that they can be aerobe or anaerobe, which means that they're with oxygen or without oxygen. And so we have here eubacteria, where that's the bacteria classified into those 2 kingdoms. And so eubacteria is the larger of the 2. Cyanobacteria, make sure you remember that. We talked about that briefly. They live together in those long chains or filaments, and they're covered with a gelatin-like substance, and it enables them to live in groups called colonies. They're a very important source for food for some organisms in lakes and ponds and oceans. And so the oxygen produced by cyanobacteria is usually is used by other aquatic organisms. I hope you reviewed over the scientific method and that you went back. You can look at your notes or have watched this video. But the main thing to know for this is being able to identify, the steps of the scientific method and what is important about the scientific method. So these were the steps. If you have not looked at this website, please do so. And so you always remember we start off with an observation. This is pretty basic, straightforward, but it's a scientific method which we've looked at for years and years throughout your various science classes. Controlled experiments. We know that in an experiment we have to have a controlled condition where something has not been changed while other things are kept constant. So we look at controlled experiments. And if you want to go, make sure you look at this section. So we know that if you look right here, it says what the key ingredients of a control is to illustrate. Let's consider this silly example, and you can go back through and read it. And they show identical pots. You have an experimental group. You have a control group. The independent variable is the amount of water that's being received. Dependent variable, which is going to be dependent, fraction of seeds that actually sprout, because that's going to be dependent upon another factor. So please make sure you know the differences between independent and dependent variable and being able to identify. The next link that I really hope you review was the scale of cells, where it showed you how to be able to calculate the cell's size. And so it also pointed out cell theory. We know the key components that went into the cell theory and the scientists that were most influential. And so we want to think back to the microscope, our last lab. And some of those very influential people would be Robert Hooke or Anton van Leeuwenhoek. So make sure that you have gone back and reviewed the cell theory. You've also looked at the microscopes and studying those. And so we have, just remember, we focused on the compound light microscope, but understanding magnification is very important. Hint, hint. So make sure you know the magnification and how magnification is calculated. A video clip has been provided for that. And that's just some plain key terms: the cell microscope and cell theory. Cell discovery and cell theory, this all ties in as well as with the microscope with Rudolf Virchow, Sheldon Swan, Leeuwenhoek. All of these, please make sure that you just have an overview of these scientists listed and what our cell theory states. There are 3 main points to know. So this right here is attacking 2 different components. You're looking at the history of the cell as well as the microscope and how it's been influential into the development of the cell theory. This Khan Academy link is going to give you that introduction to the cell, basically what I've already talked about before. And so it's going to talk about, your basic cell structure, practices identifying the cell, and understanding cilia, flagella, and pseudopodia. Remember, pseudo means false. So that's something you may just want to make sure you review over. But the key thing is understanding that cell. Here's another link that was provided to you on taxonomy. And so it really gives you some different information that can help you. And so make sure that if you are still lost on taxonomy, being able to understand the naming system, review this link as well. This is from the last lab that we did.
The natural environment teems with microorganisms. Every ecological niche provides a specific combination of nutrients and oxygen that allows only certain bacteria to survive. We can use bacteria from pond mud to create and study a mini ecosystem called a Winogradsky column in the laboratory. The Winogradsky column enriches for the mud's anaerobic microorganisms.
In wrapping up, make sure that you go back and review biological macromolecules. Remember, monomers are the building blocks for larger molecules known as polymers. and that we studied carbohydrates, lipids, and proteins. Nucleic acids is part of that, but we focus on these three. Make sure you know this chart and that you're able to utilize or understand which tests correlate to the responding macromolecule for identifying